So this lecture is a continuation of what we covered for this week, which is sensation and perception with the focus on the visual system. This is lecture 2, and we'll be talking about visual perception. So we're going to go into the brain and discuss the ways in which the brain allows us to perceive specific characteristics of our visual world. So we ended talking about the conversion of light into a neural impulse, and we were in the retina. So information from our brain enters the optic nerve, shown here, and then crosses up the optic chiasm, but more specifically just the inner halves of the retina, shown here and here, end up crossing at the optic chiasm and enter the opposite sides of the brain. So what you might notice here partially is that the visual system is both inverted and right and left reversed. So as I showed you, information from the left visual field ends up on the right hemisphere in the occipital lobe, and information from the right visual field ends up on the left occipital lobe. So that's not information from the eyes that ends up on opposite sides, but rather what's outside in the visual field, what the eyes see that ends up on the opposite sides of the field. So as I mentioned, the reason why, or the reason how this occurs, is because information from the inner halves of each retina, shown here, end up exiting through the optic nerve but crossing at the optic chiasm, while the information from the outer halves of the retina stay on the same side that they originate. So from the optic chiasm, the information enters the optic tract, shown here. So this is the optic tract. And then synapses with the first stop in the thalamus. Remember, the thalamus is the relay center of the brain. So the first stop for the visual system is in the thalamus in a structure called a lateral geniculate nucleus, or LGN. I'll be abbreviating it LGN. From the LGN, white matter tracts called the optic radiations, which you can't see here, the visual information heads to the primary visual cortex. The primary visual cortex is in the occipital lobe, which largely just handles visual processing. Interestingly, there's a disproportionately large area of the occipital lobe that processes information from the fovea. But remember, that shouldn't be too surprising, because the fovea is completely densely filled with cones, the photoreceptor that's responsible for gathering information about detail. So because so much detail is coming from the fovea, the brain sets aside more space to process this information. So let's go to the LGN. So remember, the LGN, or lateral geniculate nucleus, is the first stop from the retina that information is further processed. There are two layers of the LGN, the parvocellular, which is the outer 4 layers, and the magnocellular, which are the inner 2 layers. So magno means large, so the cell bodies of the magnocellular neurons are larger. Parvo means small, and so the cell bodies of the outer 4 layers are much smaller. So the parvocellular layer handles the what, so perception or identification of objects in our visual world. The inner 2 layers, which are the magnocellular, handle the where, where objects are located in reference to where you are in the visual world. Finally, sandwiched between the 6 layers of the magno and parvocellular are the conicellular neurons, and these guys are in yellow here. They're kind of sandwiched here in between each layer. These neurons seem to handle information about color. the differentiated layers of the LGN allow for the efficient delivery of relevant visual information to the primary visual cortex, or V1. And then ultimately from here, information goes to the secondary visual cortex. But in the previous slide, I talked about the optic radiations. Here they are. Remember, the optic radiations are the ones that connect the LGN to the primary visual cortex, or V1. So let's discuss the ways in which specific visual details are processed from our visual world. So we're going to focus on 4 different types of visual details, and there's many more that we won't discuss in this course. There's contrast, edges, movement, and then end points of objects, where objects end. So contrast is processed by specialized cells in the retina. These retinal ganglion cells are called on and off center cells. They respond to light and dark in our visual world, and they allow us to see contrast. We'll discuss this more in a second. Edges are detected by specialized cortical cells known as simple cells. They also respond to orientation of objects in our visual world. Movement is detected by complex cells, which are also found in the primary visual cortex. So these cells have tons of action potentials when objects move across our visual field. And lastly, there are hypercomplex cells in our cortex as well that respond best when there are endpoints of objects that are in our visual field. So let's talk about the contrast first as we move through the different visual details in the next few slides. So what you're looking at here in these circles, in these 2 concentric circles, is the receptive field or visual field for on-center cells and off-center cells. The reason why they're called on-center cells is when the inside of their visual field gets lit or is brighter than the outside, the on-center cell has an action potential. So anything above this line shows activity, anything below this line is hyperpolarization or inhibition. So when light hits the center of an on-center cell, it causes this cell to become really activated. On the other hand, if light hits the outside, the outer circle or the surround, it causes the cell to become inhibited or hyperpolarized. Right? So on-center cells become activated or turn on when the center of their visual field is brighter than the surrounding portions of their visual field. That's why they're called on-center cell. And they respond in opposite ways to light hitting the center versus the outer circle of their receptive field. So off-center cells, I'm hoping you guessed, they're called off-center cell because when light hits the center of an off-center cell, they shut off. They're inhibited. The opposite happens when light hits the outside of the receptive field or the surround. The off-center cell becomes activated. So these cells, because they're responding in such discrete, specific ways to light hitting the center versus the surround, they are responsible for detecting contrast, areas that are brighter versus areas that are dimmer. And it's the actions of these cells, on-center and off-center cells, that allows us to see this illusion known as Mach bands that we'll talk about later in this lecture, but not just yet. Oh, never mind. We'll talk about it next. And it's the responsibility of these on-center and off-center cells that allow us to see the illusion known as the Mach bands. So I don't know if you can see this on the screen, but if you go to the area where one band transitions into the other, it appears that the band The next band is lighter closer to this portion and is darker over here. Same thing, if you look at this band, this area of the band appears lighter compared to this band, when in fact the shading of gray is identical throughout each band. So the reason why, right, information about edges and contrast is perceived through a process known as lateral inhibition, and information about edges and contrast are sent from these on-center and off-center cells to our brain. So the way lateral inhibition works is that neighboring cells, when a neighboring cell's neurons inhibits the activity of a particular cell, enhancing the perception of contrast. So remember the horizontal cells that we really didn't spend much time talking about? So as the horizontal cell's inhibitory effect expands across neighboring cells that were not as excited as the original bipolar cell, the effect is that they're more strongly inhibited by the originally activated bipolar cells. So those horizontal cells that are transmitting information across bipolar cells, when you have one bipolar cell activated, right, if you're looking at the cell, when you have a bipolar cell that's activated, there's a horizontal cell that's shutting off the bipolar cell that might be seeing or responding to this portion of that particular this particular gray band. So what the brain perceives is that it perceives a difference in shading between these two areas when there really isn't, but that's largely because of how the horizontal cells are inhibiting one area of the bipolar cells compared to the other. So if you don't get this, totally fine. I'm going to erase these marks so that you can get a better visual of the Mach bands, because I didn't give you a chance to really look at these Mach bands. But don't worry about how you're able to view this illusion that the Mach bands appear lighter closer as they get closer and transition to the neighboring gray bar, but they do as a result of something called lateral inhibition. So now that we're done, hopefully not confusing you completely on contrast, the only thing I want you to take away from this is that on-center and off-center cells in our retina allow us to see contrast. So let's transition into how our brain processes edges and movement. So in our cortex, in our primary visual cortex, we've got specialized cells, simple, complex, and hypercomplex, that respond to different features of our visual world. So here's a subject looking at a cell phone and I think a razor. Each of these objects has an edge and an orientation to it that our retina detects, our retina and LGN detects, and then sends this information to simple, complex, and hypercomplex cells. So simple cells are responsible for detecting edges. They respond to straight lines and objects in particular orientation. So there are types of cells that respond to the orientation that looks like this, another to an orientation that looks like this. These neurons in the primary visual cortex get activated, they fire, they have action potentials, and then they send it to the complex cell that responds to any movement of these objects in this orientation. Hypercomplex cells detect where these objects end, the end points of the cell phone or the end points of the razor. They're responsible for detecting that. So in this way, these different types of cells in the primary visual cortex, even though they're processing different characteristics of our visual world, they're sending information to the secondary or extrastriate cortex and then further higher processing centers of our brain, allowing us to view these objects as wholes as opposed to just by their edges and movement. So in addition to the primary visual cortex, there are other areas of the visual cortex that are associated with specific visual functions. So in monkeys and fMRI data in humans, what they found is that cells in the secondary visual cortex, known as V2, respond to outlines and defining shapes, including illusory contours such as this one that don't actually exist. So I'm sure you're seeing that there is a whole triangle here, even though there are absolutely no lines connecting any portion of this triangle, but our brain perceives this because there are cells in our secondary visual cortex in V2 that get activated in response to illusions such as this one. So the cells of the V2 generate a perceived visual edge, the triangle, even in the absence of any visual outline that exists in this particular image, which is pretty cool. Areas in the V3, so just to orient you, V1 is located in the centralmost portion of the occipital lobe. V2 is located just in the area surrounding V1. But V3 cells, shown here, which surround V2, are involved in form analysis and motion. They facilitate our interpretation of fast-moving visual stimuli, such as a car speeding through a stop sign. Cells in the V4, shown here, facilitate our ability to identify objects as well as color perception. So information from the simple and complex cells gets sent to the V4 that allow us to see whole objects and determine what they are. V5, or also known as the MT, contributes to the perception of motion. If anybody were to have damage to this area of the brain, for example, it would make it seem like someone is viewing the world in still frame pictures instead of seamless motion. So if you happen to see that car going through that stop sign, you would see it in very jagged images as opposed to a smooth transition of that car blowing through that stop sign. Hopefully you didn't do that or you didn't view that, but that would kind of be the image that your brain would see if your V5 or MT were to be damaged. One thing that's kind of critical and important to us humans is our ability to view and detect people's faces. So the fusiform face area is a really important structure for visual information, but as it relates to face perceptions. So humans are really, really good at detecting faces. So I just want to orient you to these two images of the brain. So this particular image of the brain is as though you were viewing the brain from the chin up, right? So this is the ventral surface of the brain. This is where the nose would be, and this is the back of the brain. Over here, this is the side view of the brain. The nose would be here, the back of the brain is here, and here's the top of the head. So the view of this particular brain is as though you were viewing the brain from this way up. Okay, so this is the ventral surface of the brain. And here's the fusiform face area I was talking about earlier. But as I was saying earlier, is that humans are really, really good at detecting faces. And many times, I don't know if this has happened to you, but I remember someone because I recognize their face, but I can't remember their name, and I don't know where I've seen them from. So if I happen to see you in the grocery store at some point or out on the street, don't be surprised if I recognize you, but I look completely confused because you're completely out of context for me, but I really am good at recognizing faces. So our ability to recognize faces is the result of multiple different brain areas: the fusiform face area, the occipital face area, as well as the STS face area. These three structures collectively contribute to our ability to recognize faces. So when you end up with damage to any one of them, but more specifically if you have damage to the fusiform face area, it would lead to a very specific disorder known as prosopagnosia, the inability to recognize faces. So these individuals perceive a face, but they can't recognize who that person really is. And sometimes that happens even to themselves when they're looking in the mirror. So in addition, the occipital face area and the STS face area are really important for facial recognition as well. Damage to the STS or the occipital face area might lead to something like this. It reduces the quality of visual information about parts of the face, eliminating the person's ability to recognize individual faces. So this image that you're seeing here, hopefully you recognize it as the Mona Lisa, is what somebody would perceive if the occipital face area were to be damaged. So for visual information to be recognized and interpreted as quickly as possible, as in the case of this frisbee here, there are specific details that are processed in the cortex of broader categorical information such as a person's surroundings. So from the V1, which is shown here, the primary visual cortex, there are two streams of analysis. The ventral stream, which is shown here in blue, extends from the parvocellular layer's processing of information from the LGN by handling the what in the visual world. Remember, the parvocellular layer of the LGN handled the what? Well, so does the ventral stream of analysis, but we're now in the cortex. Similarly, the dorsal stream of analysis handles the magnocellular's processing of the where objects are in the visual world. So the dorsal stream is this area here. It's information that gets sent from the primary visual cortex to areas of the parietal lobe. So these two streams of analysis allow us to detect that this is a frisbee, and, well, it's not going to hit me in the face, but it's moving past my face. So these two streams of analysis are important for the what and where, and extending or further processing information from the parvocellular and magnocellular layers of the LGN. So around 1800, British physicist Thomas Young, shown here, proposed that there were three different receptors maximally sensitive to the perception of blue, green, and red. What's amazing is that he came up with this theory even before they were able to identify that in our retina there are three different types of cones. But he correctly predicted that one was sensitive to short waves, the blue cone, one was sensitive to medium waves, the green cone, and one to longer wavelengths, the red cone. 50 years later, Helmholtz, a German physician, wrote about the role of cones in color perception. Because they both came up with a similar theory, this theory came to be known as the Young-Helmholtz trichromatic theory of color vision. It essentially states that our ability to see color is the result of activation of three sets of cones, activating at various levels, allowing us to perceive all colors of the visual spectrum. In the late 19th century, Ewald Hering proposed a different theory to explain our basis for color vision. So he theorized that the visual system was designated to detect opposite pairs of colors, specifically red and green, blue versus yellow, and black versus white. This theory came to be known as the opponent processing theory of color vision, and it's supported by the after images that are generated from visual demonstrations that I'll show you in the next couple of slides. But essentially what happens with these after images is that the opposite color is perceived after staring at one particular image as a result of fatigue, because receptors in the eye become overstimulated for opposing colors, and so when the image is gone, we end up activating the opposite color. If you want to know more about how this works, I'm happy to share it with you, but let's do a demonstration of the opponent processing theory of color vision. So before we do the demo, let me tell you about it. So both theories are essentially correct to some extent at different levels of the visual system. So the cones, the three cones, represent the trichromatic coding theory. And the retinal ganglion cells, they're actually retinal ganglion cells that respond in opposite ways to complementary colors, red and green, blue and yellow, and black and white. So let's do this demonstration. So you're going to stare at this red dot here in the middle for about 10 seconds, and I'm going to change the slide, and hopefully you will see a demonstration of the opponent processing theory. So ready, go. So hopefully you saw the American flag, which is what you're supposed to see. So the 3 cones that are responsible for detecting the different wavelengths of light are blue, green, and this should be red, but I'm not sure why this is actually colored purple. But it actually detects red wavelengths. And so these colors allow a combination of activation allows us to see all the colors of the visual spectrum, and that's what the trichromatic coding theory essentially states. So we might wonder why well, sorry, hopefully you heard, or ignore that, that's my cat. So many people have theorized for the reason why we see in color. Like what's the reason that we need to see in color, besides the fact that it makes our world really, really beautiful? But the perception of color might actually have many survival advantages. Specifically, rods and cone ratios have been selected for specific habitats. So not all mammals see the world that way we do. So for example, squirrels and rats are dichromats, so they actually only have 2 cones, but because a squirrel happens to be an animal that is more awake during the day, diurnal, it relies on color, and so it's got 86% of cones in its retina. The rat, on the other hand, is a nocturnal animal, more awake at night, so its retina only contains 1% of cones. So this illustrates that the ratios of cones to rods have been selected for various habitats. But what's incredible is that primates are the only mammals that have 3 kinds of cones. What may have been the advantage for us to be able to see the world in so many different colors? So if we didn't have color perception, this is what we would see in our visual world. This black and white, boring, nothing, right? Our ancestors may have seen that. And so seeing this image, you might also miss critical information that color provides. So in this image, you might see that, holy cow, there's a panther there that I might be lunch for, and you would be able to run away. But in this previous image that's black and white, the panther is completely obscured and we're unable to see it, right? So color image or color vision allows us to see objects of different colors against different colored backgrounds, so that we might be able to detect possible predators that are out to get us. In addition, it allows us to discern food that's ripe versus not, find the food in a sea of green that might be more ripe. So I was just out picking tomatoes this past weekend with my family, and I wouldn't be able to tell which ones were tomatoes and which ones weren't, or which ones were leaves, if I couldn't see in black and white, because everything would appear gray to me. But the red of the tomatoes really stood out against the green of the leaves, so I was able to easily find not only the tomatoes, but the more ripe tomatoes. So the advantages of having color vision clearly have provided our ancestors survival advantages over those that didn't have it, pressuring the evolution of color vision than not. So that's it for this lecture. Let's summarize the information. So we've covered a lot of information in this particular lecture. So information from one visual field ends up on the opposite side of the brain through the optic chiasm, but remember, it's just the inner halves of the retina, the information that crosses. The outer halves of the retina stays on the same side as it ends up on the right or left hemisphere. From the eyes, visual information synapses onto the LGN, lateral geniculate nucleus in the thalamus, where different layers, parvo, magno, and conicellular, process different types of visual information. Visual details such as contrasts, edges, and movements, as well as endpoints, are processed by specialized cells in the retina or the brain. So contrast, remember, was processed by those on-center and off-center cells in the retina, but then you have edges and movement and endpoints detected by the simple, complex, and hypercomplex cells. Specific cortical structures in the occipital lobe and surrounding areas are responsible for analyzing characteristics such as form, motion, and color perception. Fusiform face area along with the STS and occipital face area work together to analyze faces, allowing us to recognize our brother, sister, friend, or mother. The dorsal stream handles the where of objects, while the ventral stream handles the what. And lastly, the trichromatic and opponent processing theories try to explain how we perceive color, which likely evolved for a number of important reasons, including being able to tell ripened fruit from unripened fruit. So that's it for this lecture. Don't forget to submit the reflection.